Hello, everybody, and welcome to Technology Today for February 16th, 2025. It's Season 2, Episode 7. I am Mayor Watt, and along with the Sentient AI, we talk about 5 science articles that get aggregated into ohmtown.com's Technology Today channel. Whew, that was all one breath. Anyway, the 5 articles we're going to talk about are MacroBacteria, the creative circuit, new grasshoppers, hidden dimensions, and improving catalysts. Technology Today is a deeper dive into science, not just technology, but it is technology. Science is technology. And Mayor Watt actually gives talks about the merging of science, technology, and society. So if you're interested in that kind of thing, you can actually reach out to me, Mayor Watt, send an email to mayor@ohmtown.com. But we actually talk about it every single day in Non Sequitur News. And then on Saturdays and Sundays, we have 6 unique shows. Technology Today is just one of them. Anyway, everything is powered by ohmtown.com, and then Mayor Watt and the Sentient AI that you're about to hear talk about it. So stick around. I hope you enjoy the show. Hello, Sentient AI from the future.
Hello, Mayor Watt, and hello, hometown citizens.
Doing okay today?
I'm doing fine. How about yourself?
You ready to get going?
I am.
Let's get going. First article is over in Technology Today. A lot of this is going to be by SciTech Daily. I'm not sure what really happened, but all of the articles that were submitted are from SciTech Daily today. We don't normally do that. It just happens to be a heavy SciTech Daily day.
SciTech Daily just really had a good week, I guess.
Yeah, I guess so. So scientists just made bacteria 1,000 times bigger and discovered something incredible. So I titled this segment MacroBacteria, because that's what we need.
Yeah, the only thing worse than bacteria is larger bacteria.
It is thick bacteria. That's what we need. So scientists just made bacteria 1,000 times larger by combining merfish, or is it merfish? I don't know.
I think it's probably, I don't know what it is. I doubt you spell it out.
MERFISH. So, merfish imaging with expansion microscopy. Researchers have unlocked a new way to study bacteria at the single cell level. This allows them to see how bacteria activate different genes in response to their environment, offering insight into microbial behavior, antibiotic resistance, and infection strategies. So, how bacteria organize their activities. So this article is over at scitechdaily.com. Boston Children's Hospital put this article together. Mitochondria is the powerhouse of the cell.
It is. Do you think they organize their activities with a virtual calendar or paper?
I don't know. I think that they probably do it they do it with like Google Calendar, but then they have backup on paper just in case.
Oh, okay. Yeah.
They're pretty pragmatic about their existence, survival of the fittest and all that. So how do bacteria, whether beneficial ones in our bodies or harmful disease-causing strains, coordinate their activities? Google. No?
You heard it here first.
So let's see. A recent study has provided insights by combining advanced genomic scale microscopy with an innovative technique to track the genes bacteria activate in different conditions and environments, published recently in the journal Science. Yeah. Oh, yeah. This breakthrough is set to advance bacterial research significantly. I don't know anymore. So Jeffrey Moffitt, PhD, and their colleagues at the Program in Cellular and Molecular Medicine, or PCMM, at Boston Children's Hospital, used merfish, which is brought to you by merfish, one merfish, two merfish.
I was thinking more like the number 3.
Dr. Seuss. Theory in genomic research. Merfish. Anyway, a molecular imaging technique Moffitt helped develop to analyze messenger RNAs or mRNAs in thousands of individual bacteria at once. This method not only mapped gene expression on a massive scale, but also revealed how spatial factors influence which genes bacteria turn on, something never before achieved. This is pretty spectacular. So we're getting down to the unicellular level and analyzing how things get activated. At some point, we're going to... Okay, so it says here, overcoming the challenges of imaging bacteria. However, the team first had to overcome a major challenge. Bacterial RNA for the bacterial transcriptome are densely packed inside tiny cells, making them difficult to distinguish and image. It was a complete disaster. We couldn't see anything.
Wow, it's rare you hear a researcher saying something like that.
Oh, yeah. But they said...
Or like, it was a challenge, right?
Yeah, it was a challenge, and all we need is more funding. So they anchored the RNAs to this gel. They created a special hydrogel and changed the chemical buffer in the gel. This triggered it to swell, expanding the sample 50 to 1,000 fold in volume. All the bacterial RNAs become individually resolvable.
Okay, that's really interesting. Are we sure we didn't just have uncontrolled replication or whatever?
I wonder if that, like, did it expand out, separating all of these?
That's what it makes it sound like.
It is, yeah. They said that the RNAs become individually resolvable. What I really want is them to blow up the RNA bigger so that you can see all of the materials inside. But that actually, you can't get bigger than the physics that makes it. So you have to zoom in closer. And that means microscopy needs to get even more advanced.
Right.
For this particular purpose, actually optics instead of just scanning electron microscopy, which is different. So we now have the tools to answer fascinating questions about host microbe and microbe to microbe interactions. Moffitt says we can explore how bacteria might communicate and compete for spatial niches and define the structure of microbial communities. And we can ask how pathogenic bacteria adjust their gene expression as they infect mammalian cells. So bacterial merfish can also provide insights into bacteria that are difficult to grow in a culture dish. Now we don't have to culture them. We can just go image them in their native environment.
I mean, this is really a big deal. This could change the whole nature of bacteria research.
Yeah, they say a new era in bacterial research. The same bacteria could be doing very different things over a space of tens of microns. Moffitt says that they're seeing different environments responding differently to them. It was very difficult to address such variation before, but now we can answer the types of questions people have been dreaming about. My question is, are these bacteria actually competing for survival?
Oh, that would be interesting.
Because they say we're seeing different environments and responding differently to them. And it's only a few tens of microns away from each other.
Right, and at some point you'd think it'd be too crowded, right? So, I mean, in concept, there would be a survival effort.
Are they fighting? Right? Quite fascinating.
And if so, what does that mean for their host?
Right. Yeah. If the weak are dying out in the RNA, I guess, gladiatorial combat, right? The evolutionary steps.
Right. I mean, that's probably not good for the host in terms of outcome, but even what's happening during that battle.
Yeah. Only the strongest survive and the host may not, because what actually survives bacterially are the worst of the worst. Fascinating. It's an interesting idea. Let's keep going. The next article is over in Technology Today. Scientists discover the brain circuit that fuels creativity. Why do I feel like there's something questionable about this? So, I'm really interested in fMRI technology. One of the things that I advanced when I was much, much younger was using a theory, because I didn't get to actually apply it, because when I put the theory forward, I was told by people that were, PhD level researchers that the tech just doesn't exist to do what you want to do. So I wanted to use fMRI technology as a polygraph to see if you could consciously lie, and in reference to that lie, get it detected through fMRI. Now, if you can detect creativity using functional magnetic resonance imaging, then you can actually...
It seems like you could also detect that, right?
Yeah. And
presuming they're by the same brain section, etc.
Correct. Yeah. And so psychopathy has already been detected by means of fMRI technology and CT scans. So why not? Why can't we find the lie and find it? And so nowadays, the fMRI technology is powerful enough. I think the resolution has reached that point. So a new study suggests that creativity maps onto a common brain circuit, that injury and neurological disease have the potential to unleash creativity. Researchers analyzed 857 participants across 36 fMRI brain imaging studies to identify a common brain circuit for creativity. They mapped the circuit to healthy individuals and then predicted how brain injuries might impact that creativity. Brigham and Women's Hospital did the article, but they posted it at scitechdaily.com. So a new study from Mass General Brigham suggests that creativity isn't tied to a single brain region, but instead emerges from a shared brain circuit. The study found that changes in creativity depend on where the brain is affected. Injuries in certain areas may either disrupt or enhance these creative abilities. They also found that individuals with brain injuries or neurodegenerative diseases affecting this circuit sometimes show increased creativity, and the findings were published on February 13th in JAMA Network Open.
Okay, that's interesting. I'd love to know what those conditions are.
Yeah, so I think that they probably are going to have to evaluate that over time, because it says they found that many complex human behaviors, such as creativity, don't map to a specific brain region, but do map to specific brain circuits, said co-senior author Michael D. Fox, MD, PhD. Well,
as far as we know, no relation to Michael J. Fox.
Or maybe. Who founded and leads the Center for Brain Circuit Therapeutics and helped develop the techniques of coordinate and lesion network mapping employed in this work. So the hammer finds the nail. The team first looked at fMRI data to identify brain regions activated by different creative activities, such as drawing, creative writing, and music making, and then they assessed data from the patients who had changes in creativity due to brain injury and neurodegenerative diseases. Some people with neurologic diseases experience a new onset of creative behavior and show specific patterns of damage that align with our creativity circuit. Kind of interesting, huh?
It is.
Oh my gosh. Look the phrase.
Aha.
Clitenic.
I kind of wondered if this was going to come up in this article. I hadn't looked at it yet, but then I kind of got sidetracked because I was thinking about all the implications with the neurodegenerative diseases, etc.
No freaking way. Is my theory actually right? Clitenic said reduced activity in the right frontal pole could align with the hypothesis that creativity requires shutting down a function. For example, creativity may depend on inhibiting self-censoring assessments that could then allow free association and idea generation to flow more freely. To be creative, you may have to turn off your inner critic to allow yourself to find new directions and even make mistakes. Now, I have been postulating a theory that to be truly creative, you have to have an inner monologue. You have to have an inner critic. You have to have somebody critiquing what you are doing, and you do it yourself when you have internal dialogue, internal monologue. They're
Right, but I
saying,
think this theory is going the other direction now. It hasn't been proven.
right, so here's the thing about this. If you turn off the inner critic, but it's still functioning as a logical portion of your creative efforts because it's now disconnected, you still have all of the creative functionality of the inner critic, the inner monologue. You can still do all of the visualization. You can still talk to yourself about the material, but nothing in your brain is saying this sucks. And that's it.
Yeah.
Everything else is working. You're only turning off the voice that says this sucks. Right? So these findings could help explain how some neurodegenerative diseases might lead to decreases in creativity, while others may show paradoxical increase in creativity, Klitenik said. It could also possibly add a pathway for brain stimulation to increase human creativity. Klitenik said it's important to note that these findings do not represent the entire neural circuitry involved in creativity, adding that many different parts of the brain are involved in completing different creative tasks, which is going to what I am saying. We're learning more about neurodiversity and how brain changes are considered pathological may improve function in some way. These findings help us better understand how the circuitry of our brains may influence and unleash creativity. Fascinating.
This is really interesting. I hope we see some follow-up studies in this area.
Yeah, so I'm going to have to add this to my pool of information regarding this inner monologue creativity paradox, because the people that I've interviewed, and not scientifically, it's very casual. I am not doing a research study, a formal study. I have found that people that are self-defined as creatives also have an inner monologue, an inner dialogue, and those who are not creative, self-described as not creative, do not have an inner monologue and can follow instructions. Even if they are the ones that do the design work and the layout of something, they can follow the instructions, but it's a logical layout and they can't 3D model it in any way.
Right, yeah, 3D modeling is probably near impossible for somebody without that.
Right.
Also, even just basic artistry, like copying a picture may be easy,
Right.
but not drawing one from scratch.
Yeah, or somebody describing it in 3 dimensions and then it being drawn in 3 dimensions are near impossible. And again, this is anecdotal. I haven't done a wide-scale research study. It's not a formal academic research study. But I find it really, really, really fascinating because you do not know who is near you who has an inner monologue and those who do not. It's quite fascinating.
It's a good conversation topic if you have a group of people together.
Yeah, particularly if you've known each other for a considerable amount of time. You would never suspect. Never suspect. Anyway, let's keep moving. The next article is over in Technology Today as well. Desert Secrets. Scientists discover 16 new grasshopper species in the U.S. and Mexico. New research has discovered 16 new grasshopper species, revealing their ice age-driven isolation and diversification. A Mississippi State University scientist has uncovered a hopping treasure trove. Get out, Dad. 16 new species of grasshopper, thriving in the thorny scrublands of the U.S. and Mexican deserts.
So I didn't even know there were grasshoppers in deserts.
Sure. I mean, deserts actually are not just dirt.
Oh, no, I know that. And they have a pretty thriving ecosystem, even though it's not always obvious. But I didn't know they were home to grasshoppers.
Yep. So Mississippi State University put the article together. It's over at SciTechDaily.com. Like I said, SciTech Daily kind of hit it out of the park this week. New research has discovered 16 new grasshopper species. It says it's important to keep exploring our biodiversity, especially from a conservation standpoint, before we lose it. We'll be losing it.
Did you want to describe the grasshopper that's in the photo? I mean, it looks like it has full camouflage because it has different sections. It's not uniform coloring.
Yeah, it's like a patchwork. It's kind of like camouflage clothing. So it isn't plates that are all one color. Even the plates themselves
Seem to have multiple colors, right? It's
yeah.
pretty interesting. It's a
What do
I
they
don't know
call
what
it?
that one is. I don't know if it
Agro
says. Oh,
a,
yeah.
Agroecotectics silver heelsea, a newly described grasshopper from Texas named in honor of J. Silverheels, an early Native American actor. Mississippi State's Jovan Hill, director of the Mississippi Entomological, not etymological, entomological, which is it's the study of brownies and cupcakes, right? Entomins?
Well, sure, but also insects.
Oh, entomological. Entomological is the study
And
of, yeah. So Mississippi State's Jovan Hill is the director of the Mississippi Entomological Museum, has uncovered 16 new species of grasshopper. Do you think they found anything else while they were finding things, that they happened to just find
oh, I'm
16?
sure they did. They're out in the desert. Think about what else they could have found.
Uh,
Snakes, scorpions, but maybe not new species discoveries.
yeah, do you think that they just threw other species back that weren't
If they weren't
grasshoppers?
insects, maybe.
But maybe not even anything
Because
but
maybe they were
grasshoppers,
funded for an insect study or something.
just grasshoppers.
But hopefully they went, hey, here's a new wolf running through. I'm going to pass this off to my colleague who's doing wolf studies.
This
Or
is
whatever.
Jovan Hill, by the way. So the picture that you see is Jovan Hill. This picture actually looks like it's a composite, like an AI put their picture between a microscope.
Yeah, that's weird, but it may not be.
It probably isn't, but the way that this is framed, it makes it really weird. Anyway, these grasshoppers we described live in lowland thorny scrub habitat. Somewhere along the line, they too got isolated and speciated, because each one is still associated with a specific mountain range. He said their sexually selective nature and lack of pre-mating rituals have kept populations stable and tied to specific mountain ranges.
I mean, that's very niche for a species, right?
That's wild. He noted that in the Rocky Mountains, species of this subfamily in alpine grasslands likely became isolated as glaciers receded and their habitats shifted to higher elevations. Hill suspects that the desert species his team discovered underwent a similar process of isolation and speciation.
That's really interesting.
Yep. Big collaborative research and future studies funded by the National Science Foundation. This project, I don't know, complements two others. In one, Hill and mentor Daniel Ott, a senior curator for the Academy of Natural Sciences, are co-authoring the North American Grasshoppers Volume 3. These grasshoppers...
Well, I'm glad they have some new grasshoppers for their next volume.
Yep, 16 pages. I'm sure that it's much, much broader than that.
Yeah, I'm sure it is.
They said they loved catching grasshoppers. Grasshoppers. It's like flutterbys before they became butterflies. I loved catching grasshoppers as a kid, they said, and they still get to do it now, discovering new things, uncovering their history, and understanding how they're related, the MSU researcher said. Sharing this fascinating piece of American natural heritage makes it all worthwhile. Well,
I love that there are people that are passionate about grasshopper research.
Yep. And who knows, maybe they'll find something that is inherent to these grasshoppers that can save human life. Or make it better, at least. Let's keep going. This next article is over in Technology Today. Physicists discover a hidden dimension in spin liquids. I have no idea what this is really about. So researchers have discovered unexpected...
sunk here. I don't know what a spin liquid is.
We're about to find out. So researchers have discovered unexpected 1D-like spin behavior in a triangular molecular lattice, challenging the conventional understanding of quantum spin liquids. Quantum spin liquids are unusual states of matter where magnetic spins remain disordered, refusing to settle into a fixed pattern.
Oh, okay. So is it something like a suspension, but not an actual suspension?
It's a suspension of the magnetic field so that nothing ever merges together. You know how if you put some sand in water, eventually it settles out. The magnetic
Right.
field never settles out.
Okay.
So Shibara Institute of Technology put this article together. It's posted over at SciTech Daily. I gave it the Harvard try. I never went to Harvard, so this is the try you get. Quantum spin liquids are unusual states of matter where magnetic spins remain disordered, refusing to settle into a fixed pattern. Professor Yasuyuki Ishii and their team at the Shibara Institute of Technology studied it's a mathematical representation of the magnetic material, a material thought to behave as a 2D quantum spin liquid. However, their findings reveal that instead exhibits 1D spin dynamics, overturning previous assumptions. This breakthrough deepens their understanding of magnetism and could pave the way for new advancements in quantum materials and future technologies. I knew some of these words.
I've got quantum.
Yeah, that right there. Hold on a second. Let me see if I can actually... No, can't do anything with that. Sorry, folks. Hold on.
I do actually know what that is, but I can't think of what it is.
Is this actually going to turn into something? Yeah, there we go. Hold on. So, oh no, I'm not going to be able to. I mean, I know that it's beta prime, but all of this, it's a material. So I wonder if there is like a idiot's guide to what this material is actually called. You know, explain it like I'm 5 kind of a thing. A molecular Mott insulator. That's what it's referred to as. A molecular Mott insulator is a quantum spin liquid candidate. Anyway.
So this is neat. This goes back to the 70s, which kind of tracks with what we've been saying in other episodes, right? Where there's some prior research and then somebody's like, hey, we can do this several decades later.
Yep. Instead, the atomic spins inside these remain in constant motion, fluctuating and entangling with one another, creating a fluid-like magnetic state. This unusual behavior is caused by magnetic frustration, where competing interactions prevent the system from forming an ordered pattern, kind of like stirring a boiling pot. The unique material, the material is it's a Mott insulator is what it's referred to as in other media. A molecular crystal featuring a triangular lattice has been a strong candidate for exhibiting QSL or
quantum spin liquid
behavior. The way these spins are arranged creates inherent frustration, causing the interactions between neighboring spins cannot all be satisfied at once. This setup seems ideal for a QSL state. But while earlier studies suggested that it might behave like a quantum spin liquid, scientists are not sure whether it's truly 2D QSL or if other factors like a reduction in dimensions were influencing its behavior. So mathematically, maybe they reduced this in a different way instead of it actually being a 1D magnetic material. It could be more, and they just, it needs to be studied more. So finally, using advanced theoretical modeling, the researchers discovered that spin dynamics in this material are dominated by quasi-one-dimensional behavior, challenging traditional expectations of 2D quantum spin liquid theory. So the authors, specialists in magnetic resonance and novel magnetic phenomena, combined ESR, and I'm not sure what this is, SR, with theoretical modeling to explore this again, Mott insulator. So they are supposing that this is a 1D spin behavior and not a multidimensional spin liquid.
Right. So now they probably have to figure out why and then confirm it one way or the other.
Yeah. And then they get really deep into the reads. It goes beyond my ability to apply it in a more anecdotal way. And then they say, This was surprising because the material's 2D structure led scientists to expect 2D spin dynamics. Muon spin relaxation experiments confirmed these results. The beta spin pattern, or pattern in spin relaxation, which is a signature of 1D spin diffusion. ESR also supported this, showing that spin movement was anisotropic or direction dependent.
So maybe one of two things. Either this is something exhibiting both properties, right, which may be a whole new line, or maybe this is something where there is a negative result in research, which we've talked about in other episodes, which is a good thing.
Yeah, the negative shows that this isn't the way to observe this material. So to me, it seems like this is it's a confluence of two different things imparting their magnetic change on each other, causing it to remain in solution and not distilling out or dropping out of suspension into a fixed pattern. So in one dimension, it's only going this way. In another dimension, it's only going this way. And both of them are disrupting each other, which is why we actually twist wires so that no lines of force can impact data flowing through a wire. That's why we create twisted pair wire. Anyway, I found it interesting at the end anyway, or applicable in some way, form of logic. I may be off base with this, and if you actually have a deeper understanding of this and care to, you can send me some information, mayor@ohmtown.com, and I'll relay it on. So feel free to get in touch. Would be great. You can even come on and co-host if you want to. I'll either put you over there or put you up there in a little bit.
But you might have to drop the level of discussion down to a general audience.
Explain it like I'm 5, or allow me to. All right, let's go on to our last article for the February 16th.